Yeah, thank you very much. Thanks to the organizers for having me. In recent years, we've seen breakthroughs in single-cell genomics that allow probing detailed molecular states of cells along the linear DNA sequence. As we know, biological function relies, however, on where these molecules are located in the physical space of the nucleus and how these molecules work together on the cellular scale, such as in phase separation phenomena or in super. In this talk, I will show how such spatial-temporal processes can be inferred from single-cell sequencing experiments. And I will demonstrate our approach giving the example of the establishment of epigenetic marks during early development. DNA methylation is a primary layer of epigenetic regulation. It affects cytosins in a CpG context, and it's mediated by so-called DNMT3 enzymes. In development, DNA methylation is first erased from the paternal and maternal genomes and then re-established between embryonic date 4 and 5 in mouse. To understand how the methylome is established, we used an in vitro model for this phase in development, where we cultured cells long-term in 2I conditions and where DNA methylation is erased. And then we release these cells to serum conditions where these enzymes, DMT3, are upregulated. Release, we then performed it by sulfide sequencing and single-cell NMT sequencing time course over roughly 60 hours, which gives us access to the methylome, to the transcriptome, and to local chromatin accessibility in single cells. DNA methylation, as you know, is regulated locally by many different factors, and consequentially, average DNA methylation was established with different rates for different functional annotations. However, we found that actually these curves differed only by a single number, such that when we rescaled time, these curves all collapsed onto a single curve. And I think in one of the earlier talks, we already have seen another example of data collapse. Now, this is what is in physics called scaling behavior, and it means that there's only a single mechanism of how DNA methylation is established genome-wide. And we, as physicists, with our equations, we can actually hope to understand something. So now this single curve followed almost exactly a simple power law with an exponent of 5/2. In the physical sciences, such power laws are hallmarks of collective or self-organization phenomena. Now to understand whether de novo methylation is coordinated in space, we calculated so-called correlation functions or covariance functions. Previous speaker called it autocorrelation functions, as a summary statistics of the arrangement of methylation marks along the DNA sequence. Roughly speaking, it gives the probability that two sites, a given distance apart, have the same methylation state. What we see is that these correlations decay much more slowly than expected by typical enzyme kinetics, shown here by the red line. In other words, DNA methylation marks are strongly correlated along the DNA sequence, reflecting potentially spatial coordination of this de novo methylation process. Now, taken together, this shows that de novo DNA methylation during development is an interesting testing ground to infer spatial temporal processes from single-cell sequencing. And this is what I'll do for the rest of the talk, to give you an overview of our approach, first using tools that we derive from quantum physics. We first infer the kinetics along the DNA sequence from single-cell data, and then map this kinetics to a description in the physical space of the nucleus. From this, we can then infer higher order or larger or mesoscopic phenomenology, such as phase separation phenomena. Now, to go a little bit into detail, as a first step, we made a general mathematical ansatz for enzyme DNA kinetics, where enzymes bind and unbind from the DNA, and if bound, they can modify the DNA, they can methylate a given position. These enzymes can also interact in general and up to no unknown ways with each other. Such interactions contain, of course, the interesting biology, and our goal is to learn them from the data. Now, the stochastic time evolution of such kinetics is described by so-called master equations. To solve such equations for general interactions between enzymes, we are able to reversely then infer the shape of such interactions from linear sequencing data. And here, which is something, of course, a complicated procedure, I just present you the result of this procedure for our experiment. What we found is that the binding rate of DNMT3 enzymes decreases with the power of 1/3 with a distance to bound sites. Now, these are the interactions. But the biological meaning of such interactions becomes clear when we calculate the total binding rate in the vicinity of a bound site. And this binding rate increases with the length of such a region to the power of 2/3. And this has a straightforward interpretation. It is the surface to volume ratio of almost any three-dimensional object, meaning that only a fraction of binding sites are accessible for further binding. Therefore, according to the data, de novo methylation relies on a positive feedback with local DNA compaction. Now, the next step to infer kinetics in physical space of the nucleus, we calculated how small length elements in physical space co-evolve with small length elements in sequence space, now, which we actually would be measured in the experiment. This leads then to a mathematical the time evolution of the concentration of methylation marks in the nucleus. And this mathematical description, again, that we can infer or derive for the experts in the room, as shown here, this resembles those descriptions that are used in phase separation phenomena, such that our prediction is that this de novo methylation process leads to the formation of condensates of methylated DNA in the nucleus. Now, to give you something more intuitive, we performed numerical computer simulations of this model that we inferred. And this computer simulation here that you see here, color denotes the concentration of local DNA methylation. And you can see how over time, hotspots if you go from bottom to top, hotspots of condensates or condensates of highly methylated DNA emerge. And we predict that the size, the size of these condensates to be in the order of 5,000 base pairs or roughly 40 nanometers. And to challenge this model, we asked whether it was capable of predicting the genomic distribution of methylation marks. And indeed, this model that we inferred from the data and that has actually 0 free parameters was indeed able to predict the genomic distribution of DNA methylation marks represented by the correlation function for a broad range of genomic features, as well as the spatial association between DNA methylation and chromatin accessibility that you see at the bottom. Now, to test our prediction of DNA methylation-dependent emergence of chromatin condensates, we analyzed single nucleus N3C sequencing data of serum embryonic stem cells, and we've indeed found an abrupt increase in mid-range contacts for genomic regions exceeding an average DNA methylation level of roughly 40%, which is, of course, fully consistent with our prediction of a phase transition and the emergence of 30 to 40 nanometer-sized condensates. Structures of strikingly similar size have recently also been observed by super-resolution microscopy, as you can see on the right-hand side. Finally, without going into much detail, we were also able to predict the genomic distribution of methylation marks in the embryos, in embryo data. And in addition, using this framework, we identified specific changes in the genomic distribution of DNA methylation marks in gene bodies that precede gene silencing at the exit from pluripotency by up to 2 days. So 2 days before we see something in the transcriptome, we can already detect changes in the methylome. So finally, to come to a conclusion, I showed you that we have a general method for the inference of emergent spatial temporal processes from single-cell sequencing. And using this method, I showed you how the establishment of the embryonic methylome relies on the interplay between chemical modifications of the DNA and dynamic chromatin structures. Finally, as a side remark, this also emphasizes what I showed here, the usefulness of analytical mathematical methods that complement the existing computational-rich computational toolbox that we have in single-cell genomics. Finally, let me thank the group of Wolf Reich at the Baber Institute and Tim Lohoff, who did these experiments, and Fabrizio Almeida, the PhD student in my group, who did this very difficult mathematics that were actually part of this that I showed were considered to be unsolvable, and he cracked it, and also the Max Planck Society and the ERC. Thanks a lot for your attention.
Thanks a lot, Stefan. We have a question from Jason Hughes. Can your approach then use linear space information to infer 3D organization, as the 3D organization would potentially determine which marks would be put on the same time, i.e. enzymes acting on DNA that are close in 3D space? The 3D organization might determine where marks are located in linear space.
Can you repeat the last part of the sentence?
So if, as 3D organization might determine where the marks are located in the linear space.
Exactly. So I think, if I understand correctly, that what I showed here is that you cannot really distinguish the 3D organization and what is happening in linear space. Because what is happening in linear space, in this example of methylation, is fundamentally determined or a product of the three-dimensional processes. Yeah, I hope I got this question correctly.
I think so.
Let's see. Arjun has a comment and question. He says, beautifully quantitative results. How might folding properties as revealed by Hi-C and others impact the results? Like if there's a loop, would you see deviation from the L to the 2/3 law?
Yes, that's a very good question. So these typical results that you get from Hi-C data typically occur on much larger spatial scales. And they would not be reflected in the DNA, in the statistically reflected in the data that I showed you, especially in the distribution of correlations in DNA methylation patterns. Yeah, so these processes exist also like other enzyme kinetics, the processivity of DMT3 enzymes and so on. They probably exist. There's lots of evidence for them, but they're statistically, so to say, not the dominant process that is going on. So they cannot explain neither this, what I showed you in the beginning, this power law with an exponent of 5/2, or the shape of these correlation functions, which is the spatial distribution of these methylation marks.
Okay, we have one more question from D. Hu. Do you think these self-organizing properties could be recapitulated in vitro?
Yes, so it depends. So I showed you some in vitro results in terms that we have cells in vitro, embryonic stem cells, cultured embryonic stem cells. If you think about in vitro, like the synthetic biochemistry experiments, these have been done partially, and they showed, actually, for example, they showed that tetranucleosomes, if you methylate them, then they stick together, they have attracting forces. So there are synthetic biochemistry experiments that go in the same direction of what I showed you, which is, of course, good because that shows that it's physically possible and not against the laws of nature, what I'm showing you.
Great.